in. We're being recorded here. Yeah, so thanks for tuning in. We're about to go on a little bit of a wild ride. So please bear with me as we move through this. So today I'm going to be telling you a little bit about my doctoral research, which is still quite new. So kind of bear with me. But here's kind of a little teaser at what's going on. All right, so a little bit about myself. So I think Robert gave a great summary of kind of who I am and what I'm all about. But I'm a snake guy at my core. So I got into this in my undergraduate degree, really interested in snakes. And at the time, the outlet for that was through a lab that worked on birds, oddly enough. I grew up in Canada. We didn't really have much of a snake community there, believe it or not. So what that meant was studying garter snakes kind of through the lens of a bird person. So I worked on phenology, I worked on natural history, movement ecology, things that are kind of easy to look at and kind of detect with your eyes, if you will. That really ignited my passion for research. I presented at a few meetings and I really fell in love with herpetology, with this field of herpetology. And that took me to Colorado. And I joined the lab of Stephen Mackesy. I worked on a lot of different kind of molecular and proteomic topics, all kind of enveloped under the kind of umbrella, if you will, of viper venoms. So generally prey resistance and looking at things like rodents, how rodents manage to resist venom. I'm now kind of taking that a step further here in my PhD, and I want to incorporate a little bit more of an evolutionary element to this. And to do this, I'm using Cobras, which are kind of a fantastic system. And I'm going to try to explain why that is in just a moment here. All right, so when I'm talking about Cobras, I'm talking about the Asiatic Cobras. And this is a relatively recent radiation of snakes. There's about a dozen species in this lineage, and they're distributed throughout Asia. So this map kind of shows that spatial distribution of these snakes. You have species in Central Asia, species in South Asia, and then species throughout Southeast Asia. And the key thing here is that these cobras are confusing. They're a bit of a conundrum. And that's because they're morphologically similar. These snakes kind of look the same. And I'll show you what that means. So if you think about a snake like an Indian cobra, right, this might be an example of Ophidian perfection, as some herpetologists might say. But this is kind of a classic cobra, right? It's a brown snake. It has kind of that beautiful binoculate hood marking, those kind of glasses that stare back at you. And this is what a lot of people might think of when they hear the word cobra. But Asia has a lot of different cobras. And some of these cobras are stark and really well marked. And this cobra right here, the Samar cobra from a couple of islands in the Philippines, demonstrates that really well, right? So very different looking snake, bright yellow, bright orange, and kind of bold black, contrasting that. But then we see other phenotypes. And one of those phenotypes is this beautiful banded phenotype. And this is a Siamese spitting cobra. And again, black, white, very thick, prominent bands, right? And these snakes all look different from one another, at least to my eye, and at least kind of from a color standpoint. But morphologically, they're not that different. If you count scales, if you measure, things like skull width and skull length, these snakes don't really look that different. And this is where we get into a lot of confusion taxonomically. Now, to make this group even more interesting, some of these cobras actually spit venom. And this is really unique. There are only 3 lineages of snakes ever that we know of that have evolved venom spitting. So one of them is this Asian cobra radiation. Now, about 6 of these species, about half of them are able to spit venom, and then half of them lack the ability to spit venom. And that's just been one more factor that's kind of confused folks that have tried to make taxonomic judgments on this group. So venom spitting. So keep this kind of in the back of your mind for a moment here. All right. Now, the cobra that I've honed in on and decided to really focus in on is this snake called the monocled cobra. And it's essentially just a nondescript brown snake. The image right here depicts a very normal looking monocled cobra. This is a brown snake, it spreads a hood, and it generally has a single monocled hood marking on the back of that hood, looking a lot like an eyepiece or an actual monocle. And the snake has a large range. In fact, it's got a huge range when we really think about it. So it extends all the way from the mountainous regions of northern India. It's distributed throughout the Indo-Gangetic Plain, throughout South Asia, and then it makes it all the way down into Indochina and Southeast Asia. So there's a lot of different biogeographic factors that play in this area. Climates are very different, and you can think ecologically, these snakes might be doing different things across this distribution. So really interesting snake from a spatial perspective. And again, going back to that taxonomy, if we zoom in on just this monocled cobra, this provides a really nice example of this taxonomic confusion. So this snake has been described through a variety of different names by a variety of different people. And various folks have attempted to bin variation in this group into a taxon. Of course, most of these taxa are completely invalid, and they're not really supported by any description that carries any weight. But these names have provided a lot of confusion in the literature. So if you're doing, let's say, a VertNet search and you're trying to find specimens that belong to a monocled cobra, there's 15 or 20 different names that you're going to have to search to find these. And these have been attributed on the basis of, a light background coloration, an extra row of scales here or there, things that don't really matter in the big picture, but justification in the moment to provide an additional name. So very, very confusing taxonomic history. And that taxonomic history has been somewhat cleaned up in recent years, but there's continued confusion. And I think, at least, a lot of this continued confusion is attributable to these variable looking snakes, these variable phenotypes. So if we think about this on a spatial scale, I've just pulled 3 images that illustrate this point quite nicely. So a snake from northern India on the left here has this beautiful, almost zebra-like phenotype, right? It's quite finely banded. It's got some very bold contrast and coloration, and it's just a very distinct looking snake. If we think about a snake maybe in peninsular Malaysia, kind of at the southern edge of that range, again, just a unicolor brown snake, nothing really distinct or noticeable about that snake, in compared to other monocled cobras. But then if we go up into Yunnan, so the northern part of the range there in China, we're again dealing with a different look. So we've got a banded snake. It's uniformly very dark. It's quite shiny looking almost. And I think a lot of this phenotypic variability is continuing to fuel this taxonomic confusion and this desire maybe to better, qualify these different lineages, if they even are different lineages. So just a lot of confusion is kind of the theme that has been in the history of this snake and continues to kind of pervade the literature. All right. Now, this confusion doesn't really just end kind of after the eye test, right? This confusion is also reflected when we do phylogenetic analyses. So this slide is a bit of a mess. I've just pulled a few trees out of the literature that illustrate this really, really nicely. So one of these here comes from a paper, and it looked at snakes in Bangladesh, and it used an outgroup of a snake from Burma and a snake from Thailand. And what they found is some pretty significant divergence here. So this might indicate that snakes on that Indo-Gangetic plain and snakes in Indochina maybe have something going on, potentially taxonomically, but certainly phylogenetically. The tree underneath that, quite nicely color-coded here, has a little bit more rigorous sampling. And you can see in this particular analysis, 4 different lineages of Cowthia kind of fall out in that tree. It's, again, a little bit of a mess, but it kind of provides a bit of a hint that there's maybe something going on beyond what we've detected. And then I think things have really picked up in recent years. And you can see the tree on the right there is really well fleshed out, and it's got a very nice sampling. And this has been the first kind of attempt to, categorize some of this diversity. And the big take home indicated in that red box there, you can see 2 very distinct lineages currently being kind of nested in that Naya Cowthia group, right? So you have our main lineage, and these are snakes, presumably from Southeast Asia, that do form that one nice cluster. But then there's that second branch that falls out near Naya Atra, which is a Chinese cobra. So there's definitely some weird, potentially lineage sorting or just cryptic speciation going on. And this has only kind of come to the forefront very recently, right? So 2019, 2020, this is all fresh material we're kind of working with here. All right. And then things happened, and things happened very, very recently. So it was about 1 semester into my PhD. So I was teaching an anatomy class when I got this news, and it kind of blew my mind. And a group from China, kind of out of the woodwork, went ahead and they described a new cobra. And it completely shocked me because we've been, sitting on these data for 20 some years, but here it is. So a group in China went ahead and they did some molecular work, and they actually had really, really good support to elevate the 12th species of Asian cobra. So they described this snake called the brown-banded cobra, which they presume to be distributed across the higher elevation regions of southern China. So very interesting, kind of illustrating how taxonomically volatile in some ways this group continues to be. All right, I'm going to pivot. So bear with me. This is our kind of first major pivot here. So we've talked a lot about phylogeny and a little bit of taxonomy. Now let's kind of swing in the venoms direction. So venoms, what are venoms? Well, venoms are a complex kind of chemical cocktail, if you will, and they're comprised of proteins, of peptides, and of enzymes, right? And these have a bunch of different potential functions for the snake that's actually using them. So, oh, we're not switching here. So one of those functions might be digestion. This is somewhat debated in the literature. There's varying degrees of support, but the general idea being here that some of these degradative compounds in a venom actually circulate in that prey item and they pre-digest that prey item, if you will. So they help that snake break down the prey item before the snake has even really eaten it. So that's one idea. There's obviously defense, right? Defense is undeniably a very important thing driving venom variation. And, you know, most importantly, of course, in these spitting cobras. So these snakes have the ability to kind of propel venom through the air at an offender. They don't even have to touch that offender, but that venom does wonders, right, in deterring someone who's bothering a cobra. So certainly defense is an important role of venom. But then the most important role, without a doubt, is this idea of prey acquisition. And this theme gets demonstrated time and time again in the literature. Daltrey et al. Have done some really nice studies back in the 90s that demonstrated it quite nicely in this Malayan pit viper. But the general idea here is if you want to eat and you want to eat effectively, your venom needs to be able to track your prey, right? So if you're eating frogs in one area and you're eating rodents in one area, you may have to upregulate the toxins that you're producing to effectively kind of subjugate or incapacitate those prey items. So prey acquisition is huge, a really important factor. All right. And this has been something that I've really kind of played with a lot and been really interested in. And a lot of my master's work kind of revolved around this. And I have some pubs right down the pipe, hopefully very soon, looking at venom resistance, which is tangential to today's talk, but a very kind of cool example of prey counteracting some of those co-evolved venoms. But venom variation. So venom variation, generally speaking, is huge. It's a very common thing, and we especially see it in these broadly distributed taxa. So this example right here comes out of my graduate study in Colorado. These are prairie rattlesnakes. So kind of a ubiquitous rattlesnake. It lives throughout the Great Plains of North America, and it's really a boring landscape when you think about it. It's a lot of flat grassland, right? To our eye, at least. But what we see in these rattlesnakes when we do some proteomic investigation is some very divergent venom phenotypes. So in the northern stretch of those grasslands, we see what we call a myotoxic phenotype. So this is a snake that bites something and that venom is actually paralytic. So it would cause, you know, that mouse or that rat to essentially lock up and to go into paralysis. Conversely, snakes in the southern kind of limit of that Great Plains, they show a proteolytic venom phenotype. So rather than being kind of overtly toxic and paralytic, this proteolytic venom is degradative. So instead of killing that mouse quickly, it kills that mouse in more of a drawn out way, if you will. And it causes that mouse to really break down a lot more than that paralytic venom. So we're really seeing divergent venom activities even across what we would assume to be kind of a boring habitat from an environmental perspective. And this theme, again, is recapitulated certainly across rattlesnakes and certainly across venomous snakes in general. We see this all over the place. All right. And of course, one of the places we see this is cobras. So if you take a dig through the literature, you'll find, you know, a laundry list of papers that kind of reach a similar endpoint to the one I'm presenting right now. But this paper right here, which was done by a Malaysian group, looked at geographic venom variation in the monocled cobra. So relatively sparse sampling, but a few snakes from Malaysia, a few snakes from Thailand, and a few snakes from Vietnam. And they kind of crudely built just venom proteomes representing the relative abundance of these different toxin families that these cobras are expressing. And the take-home message from these pie charts is just that at the interpopulational level, so we're talking about snakes in different countries, right? 3 different populations in 3 different countries, we see really, really strong signals of venom variation. So if you zoom in on just one toxin, let's say this dark blue part of the pie chart, the LNTX, this is a neurotoxin. So, you know, interferes with neurotransmission. We can see dramatically different kind of regimes of expression in these 3 populations of cobra. So very low expression in Malaysia, relatively high expression in Thailand, and then apparently no expression in Vietnam. And that same theme is kind of carried on in these other toxins, a lot of variability. And this can cause snakebite to manifest different symptoms, right? Whether that be in a human or in a prey item. So lots of potentially interpopulational level venom variation. So that's kind of a big thing, and we see that in other snakes. But monocled cobras get a level more interesting. Because if you think about interpopulational venom variation, that makes sense, right? That would imply local adaptation. There's a spatial gradient going on. Intuitively, we can understand that. But what we don't see in other snakes is this intra-populational level venom variation. So intra being within the same population. And this is a paper out of India, West Bengal, in fact, so extreme eastern India. And what they did here is they took several cobras from one population. So these are snakes that are all in reproductive connectivity with each other. They know each other, if you will. These are snakes that live in the same area. And when they went ahead and constructed venom proteomes for this, we find that same level of variation kind of recapitulated here. So these are both snakes from the same area in West Bengal. But when we pick a toxin, let's just say this C three-finger toxin, this red color on the pie chart here, we see dramatically different expression, right? So 25% expression in one individual, 6% expression in another. So this is amazing, and it's also terrifying, right? Because it means that a monocled cobra is not a monocled cobra is not a monocled cobra in any one given area of the world. So you could be bitten by either of these snakes in the same place and get totally different symptoms as a result of this venom variation. So really, really cool sharp left in this whole monocled cobra system that we don't see elsewhere. So just kind of keep that in mind. So with all of this in mind, right, all of this phylogenetic stuff and all of this venom stuff, there's a couple of questions that really have me scratching my head and that kind of keep me up at night and motivate me to play with these cobras. And those questions are as follows. So what are the patterns of lineage diversification and trait diversification that are actually present in the monocled cobra? So not just speculative, not just looking at pictures of snakes. Let's do some actual work and find out what's going on with this group of snakes. And then secondly, how does venom phenotype, right? So the expression of those toxins, how does that vary across a spatial scale in the monocled cobra? So between populations and across the landscape, are there any patterns that we can detect in what these venom phenotypes look like? But then we have a problem, right? And the problem is that to do any of this work, you need A, genetic samples, and you need B, venom samples. And unfortunately, with these being an Asian snake, with permitting already being logistically difficult, and with CITES being a thing that these snakes are regulated under, there's next to nothing available for pre-existing material in museum collections. There's really only a handful of samples already in the Western world. So that makes things complicated. So what we did is we went and we got some new stuff. And I just want to go ahead and thank SSE right off the bat for that wonderful $2,500 award. That was very helpful in facilitating this first field season. And I could not have done this without that funding support. So a huge thanks just right off the bat. So we started off in Thailand. And this is just kind of a quick summary of my very first field season there. So the image on the left, this is, I think, day one, or at least week one. This is a still optimistic field biologist. These four people are all smiling. Things have not really started yet, so we're all happy and full of hope and joy. But that's obviously myself, my American colleague Reed McClure, who was wonderful and came out as a field assistant with me, and then my two local collaborators, so Sunchai Makchai and Michael Koda, both of which are associated with the National Museum of Thailand. So this was kind of Team Cobra for the field season, and we all did different things, and it ended up working out really, really nicely. But obviously the first logical starting place is kind of deep in the bowels of a natural history collection. So that's what we did. So that first week, we spent that week inhaling ethanol and rifling through old jars and just really finding out what the historical kind of aspect of cobras in Thailand looks like. So there you can see me very kind of joyfully hovering over a box, or a couple boxes, I guess, of cobras and ethanol. And this represented about half of the holdings that you're looking at right there for monocled cobras in that Thai museum. All right. And what that actually looks like, it's not all, smiling and taking pictures. It is a lot of hard work, and it's a lot of, at times, really boring and painful work. So we ended up doing a ton of scale counting. So a lot of just preliminary data. So are there any trends in these, features of scalation? Do cobras look any one way or another in these particular regions of Thailand? What material can we really glean just looking at some of these potentially 100-year-old specimens? So a lot of really nitty-gritty kind of fine scale data collection on features that may or may not matter, which is unfortunately just how this goes. In line with that, we also took some tissues. So for every specimen with verifiable locality data and that appeared to be in good shape, we went ahead and took a section of liver tissue. And I'm going to go ahead and try to sequence these using some relatively new methods to get DNA out of museum specimens. So definitely not a waste of time. We'll see how well these tissues perform under our protocols, but potentially a very good source of data, even if it is fragmented data or a lot of it doesn't work. So fingers crossed that that stuff works out. We haven't quite made it that far yet. All right. But you can't spend all day in a museum. Eventually you get tired of doing that and your nose can really only handle so much ethanol burn. So we went ahead and we tried to get fresh cobras. So here we had about 7 weeks and we did a long field season of what we would call herping. So just going out and looking for herps. Herps are reptiles and amphibians. So this takes you to a lot of really interesting places. So these are just, this will be a photo montage here. So just bear with me as I storytell for a moment. So we started in Bangkok. It's very easy pickings in Bangkok. So our first herp of the trip was this, beautiful water monitor. He was sharing a dead catfish with a crow in a city park. So not the most picturesque of scenes, but for a North American who has never seen a big water monitor in the wild, it's a pretty exciting find, right? All right. Things kind of slowly escalated from there on. Here's a monkey that stole a bag of chips. So we're kind of trending in a better direction, if you will. I don't know if this is better, but it's certainly entertaining. And then the herps started to pour out. We finally realized what we were doing and we figured things out and we got good at it. So we found this beautiful pit viper. This is a Malayan pit viper. It's very similar to the North American copperhead in a lot of ways. And these were an abundant snake throughout the trip. Very medically important snake. It's got a very degradative venom. Not a great thing to get bit by, but beautiful. And it was encouraging to be finally finding snakes and finding venomous snakes at that. All right. And then things really started to heat up. Here's a Wagler's viper from southern Thailand. So a beautiful arboreal tree viper. These things eat birds. They're very big and just a really satisfying animal to get to interact with. All right. The vipers really kind of kept coming, if you will. So here's some Saba pit vipers. These are again from southern Thailand. So the snake on the left, that tiny little guy, that's actually a male. He's got some beautiful red banding. This is a sexually dimorphic species. And then that big beast on the right, that's a mature female. So these snakes were found, I want to say maybe 5 or 6 feet apart. The male was presumably kind of on his way to mate with that female. And we kind of briefly interrupted them for a photo op, but I assure you they carried on business right after we left. So we're starting to feel really good. Things are happening. Elephants. Elephants were a novelty to begin with. But they very quickly became a problem. So I'm out there for 8 weeks handling all these venomous snakes. And I would say the only dangerous animal encounter I had was actually an elephant. So we were out hiking one night. I say we, but it was just myself. And I was charged by an elephant, which was one of the scariest things in the world. I didn't really know what to do. I kind of treated it like a bear. So I just turned off my headlamp and laid down, and I think it lost interest. But again, right, you can play with venomous snakes all day and you finally get taken out by an elephant. What an unfair reality. But such is life, I guess, doing field work in Southeast Asia. So watch out for elephants is my take-home message there. All right, but back to snakes. Things kept going quite well. Here's a beautiful reticulated python that we found in this really, really pretty riverine habitat. This is a young snake. It was coiled up under a bridge, much like you might find a rat snake in the US. And it was hunting bats at night, which was super cool to watch. So just a really, really interesting little natural history tidbit. All right, we took a quick meander up north to Laos because why not? We were close enough. And we were very lucky to grab dinner with a local herpetologist. So this is Dr. Samputon Pimachak, who's kind of one of the leading herpetologists in Laos, producing a ton of really, really cool work. So it was great to interact with her and build a little bit of a connection there. And of course, while in Laos, look for cool Laotian snakes. So this might be one of the gems of Laos. This is a snake called it's got a terrible common name. I believe it's called the 3-scale horned pit viper. But to kind of break things down, this snake is an absolute dragon. It lives in these beautiful high elevation limestone kind of foothills of the mountains. And it is functionally a dragon. It's got these wonderful horns over its eyes. It gets really, really big. It looks like something out of a fairy tale. This is without a doubt one of the coolest animals I've ever been able to lay eyes on. So just a really, really cool little experience. And then cobras finally started to happen. So here's a black spitting cobra. This is from just south of Bangkok, I guess southwest of Bangkok. So it's a cobra, it's a spitting cobra, but unfortunately, it's the wrong cobra. It's not a monocled cobra. So while it was really exciting, it was somewhat disappointing for this to be the cobra that we found rather than the cobra we were looking for. And then we found more cobras. Again, the wrong cobra. So this is a king cobra. People would go nuts seeing a snake like this. And of course, as a snake nerd, I did go nuts. But again, the wrong cobra, right? So in this case, 10 feet of cobra, 10 feet of king cobra, but it was not the monocled cobra that we were after. So I was beginning to get a little bit worried because we're spending all of this time herping and we're finding really, really cool snakes, but we're not finding the one snake that I'm after. And my $2,500 are slowly being expended on all of these snakes that aren't really furthering my own research program. So panic is ensuing at this point. But things took a turn. Unfortunately, we're in Southeast Asia. And this is my one wise quote that I'm going to stick with, and I'm going to keep parading for the rest of this program. But one man's curry is another man's doctoral dissertation. And it turns out that cobras are delicious. We didn't get the opportunity to eat any, but they are, a common food item in a lot of Southeast Asia. So we really lucked out interfacing with a lot of local snake catchers, and we got snakes this way. These were snakes that were destined for the food market, and we managed to kind of circumvent that and get access to these for my research, which was wonderful. And then, of course, cobras are one of those things that wander into people's houses. So getting calls from locals and getting calls from snake catchers was not an uncommon situation. This image here is a toolbox that we bought at a local hardware store, and we actually ran over to a snake catcher's and picked up 5 cobras. And these cobras all had locality information. He had, time and date of capture. He could give me an address. So snake catchers are a wonderful resource, and folks were generally very cooperative and happy to help out with this type of research. So in short, we generally did a pretty bad job at finding our own cobras, but people really, really did a good job in kind of filling these gaps for us, which I'm incredibly thankful for. And then one of the unfortunate realities of being a cobra in the wild is that you're an active snake, right? And snakes love roads, cobras love roads, cars hit snakes. It's just the reality. So it happened to be baby season toward the tail end of our trip, which worked out wonderfully because we were doing a lot of driving. And consequently, we ended up collecting a ton of fresh roadkill cobras. And these were great. A lot of these were still salvageable for venoms. If their head isn't smashed, you can still express venom from the glands. And certainly tissues are great from a fresh roadkill cobra. So we did really well. And in short, things took a turn for the best, fortunately. And what that meant is we acquired 68 brand new fresh cobra tissues, which is essentially unheard of. That's really, really great sampling. I did not expect this. Combined with that, we had 40 historic tissues from that museum. So these are going to be touch and go, but let's hope for the best. And then we had 39 fresh venom samples throughout that range. So we've really filled out the tie range of this cobra, I think, phenomenally. Of course, that northeast portion looks pretty barren, but jury's out on if there's even monocled cobras there. There certainly are in a few areas, but they're not really common in that northeast. So let's just not worry about that for now. All right. And what that looks like when we incorporate samples already in the US and samples that I already have in hand, we're starting to see this distribution filling out. So a good chunk of Myanmar is well represented, and we have a few from Indochina, but more work to be done. All right. So I'm going to address 2 questions. This is a little bit of preliminary data. It's not really that well worked out. So please kind of bear with me. So question number 1. So I talked about spitting a little bit earlier in cobras. So here's a question that still kind of plagues us today. So is the monocled cobra a spitting cobra? We don't really have a good answer for that. So here's a really great paper that came out in the 90s. This is by Wooster and Thorpe. And they tried to bin these cobras into spitting and non-spitting categories. And you can see just a quick schematic of what a spitting and non-spitting fang look like. The take home is a spitting snake has a really tight fang orifice, right? A really tight opening in that fang to allow that venom to project in a stream, whereas a non-spitting cobra has a long fang. And generally, monocled cobras appear to be a non-spitting cobra based on their fang morphology. But various people have hinted at these things spitting. And a paper came out a while back, kind of documenting this, actually putting this in writing for the first time. And what we see is that these snakes from West Bengal, these monocled cobras from West Bengal, spit. They always spit. They're spitting cobras. Then a couple more publications came out. One from Assam, so further east in India, also documented spitting in their population of monocled cobras. And then a group from Nepal did the same. They documented spitting in their population of monocled cobras. So what we're seeing is, hey, maybe these fangs don't appear to be adapted, but it does appear that at least some populations of monocled cobra are spitting. And what that looks like when we kind of map out where these are all falling, we seem to see a trend where these snakes in South Asia, kind of on that Indo-Gangetic plain, do appear to be spitting with some level of regularity, whereas these snakes in Southeast Asia don't spit. There's anecdotes of them spitting, but no one's actually kind of really come up with a good example of spitting in these Southeast Asian ones. So let's think about those FANG data. So I've managed to get that data set from Worcester, from that paper done in the 90s, and I kind of played with it using a few newer techniques. So this is just a PCA looking at 3 different metrics of that FANG. So FANG opening, FANG length, and then a different type of FANG length measure. And we see basically a mess, but we do see a trend of things kind of moving up this gradient. And you can see, well, I don't know if you can see, but spitting essentially is represented at one end of this gradient and non-spitting at the other. So at least in these variables that we've looked at, there appears to be somewhat of a clinal distribution, but the gist is that things look really, really messy the way they're depicted here. So maybe some specialization, certainly in the spitting cobras, and then differentiation in the non-spitting cobras. But when we think about just monocled cobras, do these South Asian snakes actually have any differentiation? In short, they don't, based off this data set. So a snake from Indo-Burma, a snake from Indochina, and a snake from the Malay Peninsula all overlap in FANG metrics. So is this an example maybe of incipient spitting? Does the monocled cobra represent kind of a gateway to this spitting behavior? Well, there's actually one article that documented the similar kind of thing going on in the Chinese cobra, where again, a non-spitting cobra, but an individual of the species held in captivity spit venom. So the idea here is that there might be some suggestion going on here that the evolution of this spitting behavior is actually preceded by a development of the behavior before the development of the FANG. So neat idea, jury's still out, needs more work. Now, question number 2. So the other thing that keeps me up is, what is the geographic distribution of this new cobra, this Nyafushi? So just to jog your memory really quickly, the snake was described, very recently from southern China, and the original description kind of posited this range map. So this is all Asian cobras. I'm going to zoom in on this new cobra, and you can see that they've gone ahead and they've kind of designated a little bit of a range here. So they have their type locality indicated here, and then they go ahead and they extend the range right to the Chinese border. Now, anyone who knows anything about biogeography and wildlife and range maps should know that animals don't really follow political lines very well. So this seemed kind of premature to just go ahead and truncate the distribution there. Shortly after, a group from Thailand actually published a really nice molecular data set, and they found evidence of this new cobra, Nyafushi, throughout many mountainous regions of Thailand. So you see it kind of distributed throughout the around the central plain in that mountain range that encompasses the central plain, whereas Kauthia, which are these black dots, seems to be a lowland species in Thailand. So if we kind of estimate or eyeball where these cobras are, this is just a rough kind of schematic of Nyafushi, kind of comprised off of what's available in the literature and maybe just a good guess. So we definitely see it in China. We definitely see it in the Thai mountains, but we don't really have verification of this snake anywhere else in Indochina, at least from a molecular or morphological level. And then there's some speculation that it may actually occur in the Cardamom Mountains here on the coast, down in Cambodia and in Thailand. But this is, again, speculative just off of images. So while we were inhaling ethanol and scale counting, we bumped into a snake labeled Kauthia that didn't really give off the vibe of Kauthia. And the snake was from an area called the Tanuk Waterfall in Trat, Thailand, which is pretty far south. So we went ahead and did some scale counts on this snake, and we found that this was actually Nyafushi. This was not Nyakauthia. And Trat is right where this red star is indicated here. And you can see that this represents a pretty significant range extension. So this confirms occurrence of the snake in the Cardamom Mountains, and it presumably confirms occurrence in this same region in Cambodia. So we went ahead and we did these diagnostic scale counts, as I mentioned. The silver bullet, kind of differentiating this Nyakauthia from this Nyafushi, is a single tiny sesame seed size scale. It's a tiny scale right under the chin. And again, that scale is perceived as a silver bullet. So we went ahead and looked for that. You can see the figure on the left just shows what that scale looks like. And then our snake of interest is just shown here on the right. And you can see, in fact, that it does have that scale. So we were able to pretty definitively call this Nyafushi and pretty definitively determine that this snake does occur in the Cardamom Mountains of Cambodia. So kind of a neat geographic leap for this snake to have occurred over. So in short, we still have a lot to learn about where these two species occur. This is really only the tip of the iceberg, but we're working on it, or at least I'm working on it. And what that means, I think, is further work in Cambodia, certainly. We know the snake is in the Cardamom Mountains. There's potential for it to be on some of these offshore islands. There's certainly been photo records that kind of check out and do look like this newly described cobra. But then the other question is, where is Kauthia? Where is Naya Kauthia, the monocled cobra, in Cambodia? Is that a lowland snake? Is it common in Cambodia? And then, of course, the same for Vietnam, right? We see a lot of mountainous areas in Vietnam. The Annamide Mountains cover a good chunk of Vietnam. Presumably, the cobras in these mountains are probably this new cobra, Nyafushi. Again, the monocled cobra is probably a lowland species. So a lot of work needs to be done here to kind of delimit the range of these two cobras in these regions. And that is hopefully on the docket for a future field season. All right, so to kind of wrap things up, just moving forward. So, you know, a big smattering of stuff going on here, but I guess in short, to actually finish and to make something out of this whole thing, these are the steps I've kind of plotted out in my head. So I'm really interested at the more macro evolutionary level at the biogeography of these Asiatic cobras. So when did they get to the places that they got? How did they get there? How did they diversify? I think understanding how this clade kind of works at a bigger evolutionary scale is going to be vital to understanding things at a more zoomed in level. I'm really interested and really excited to do these DNA extractions from tissues from preserved specimens. So this will be very interesting. It'll be fun to play with for sure. And there's obviously the potential here to unlock data that have never really been conventionally available. So very exciting development. With this, I'd like to do some distribution-wide phylogenomics via a relatively new probe set. So again, this will be kind of a new shot in the dark. So we'll see how this all goes. And then finally, I'm very, very curious to do some molecular fingerprinting of these individual venoms, just generally via proteomic techniques to find out what toxins are where and what's being, you know, upregulated or downregulated on a spatial scale. So a lot of these kind of topics are just in limbo right now. I'm waiting on a lot of specimens to come back from Thailand. With this being a CITES listed species, there's an additional level of complication there. But fingers crossed those cobras make it in safe and sound, and hopefully very quickly, so I can start doing some of this rather than just talking about it. Then of course, right, more field work is needed. I've only really scratched the surface of where these cobras actually occur. So my really, really big sampling priorities here are, of course, Indochina. So just trying to work out what's where. And then South Asia. There's really just a scarcity of data for South Asia. And I think there's a lot of really unique things potentially going on with those South Asian cobras. All right, so we're kind of wrapping up for time here. With that, I just want to give many, many thanks. So there were so many individuals who helped me out in the field, who really made, you know, what should have been an impossible field season just work out flawlessly. I could not have collected, you know, this amount of cobras without cooperation from all of these wonderful people. And, you know, these folks were just helpful all along the way. It was great to have such a supportive team. So thanks to everybody for that. And of course, this work would have been impossible without funding. So for that, you know, my greatest thanks go to SSE. This grant was immensely helpful for this work. I also want to thank the Animal Behavior Society for some funding, as well as the Brian P. Glass Zoology Fellowship through my own institution. So this work, you know, it took a huge team, and I'm just immensely thankful for everybody who made this possible. So with that, I'd be happy to take any questions, and thanks so much for listening to my talk.
All right, great. Thanks so much, Neil, for the great talk. Lots of fantastic images. I enjoyed seeing the field work. Yeah, any questions from the audience? You can unmute yourself or turn on your screen or type it in the chat. Happy to read it out. So if not, I'll start with so can you find some sort of genomic feature or like something related to the venom genes or gene pathway that associates with a particular venom type, even though they're in the same population, right?
Yeah, you certainly could. So a lot of groups have taken, kind of a more genomics or potentially transcriptomics angle on things. And I think that's definitely a very useful level of organization to look at things. I think my interests lie a little bit more in the functional end of things. So I'm really interested in characterizing these toxin abundances as they're expressed. So, maybe a bit beyond my pay grade, if you will, looking at transcriptomes. But it would certainly be a very interesting avenue to go down, and something you could definitely do if you wanted to.
Yeah, so yeah, that's cool. So and when you find out the abundance or the measurements of the venom, you would be able to infer the prey or what could you learn ecologically?
Yeah, so that's a great question. So it's venom is a very messy thing to play with, I think is what I've learned. And it's tough to infer, directly what's going on at a very kind of causative level. But I mean, having that information is certainly useful in the context of environmental variables, in biotic variables, and things like that. And it's certainly useful from a predictive standpoint, as far as snakebite goes. So I think that's maybe the most targetable application that you can kind of chase right away.
Cool, yeah, and I think you could learn something from the from the genomes. Not genome wouldn't necessarily need transcriptome data to just see if there's mutations or substitutions associated with certain venom types. But yeah, thanks a lot. There's some questions in the chat I'll get to. So Gregory asks, did the snakes from the food markets have good locality data? That's a good
Yeah, so
question.
that's a great question. So it's
Yeah.
it's touch and go, right? A lot of it kind of depends on your market. I know that that sounds kind of flippant, but in general, you can actually reach out to the person who caught the snake, if you have enough time in that given area, right? You can kind of follow the breadcrumb trail and find who caught the snake. So usually, yes. With that said, at some of these markets, there are a lot of snakes, and they're not really keeping tabs on what's what. So you can generally constrain things to a village or to a region. But yeah, you certainly do have to be careful with your locality data when you're acquiring animals in that way.
And Anil asks, you mentioned that many of the snakes you collected are juveniles, roadkill. Does venom composition change ontogenetically? I thought I heard it does in some rattlesnakes.
Yeah, so that's a great question. So it definitely does change ontogenetically. And there's been a fair bit of work already in cobras and in monocled cobras. My master's advisor actually published some work on monocled cobra venom ontogeny. So that's certainly something that would be great to check out. But yeah, that is definitely a problem, right? You have these
Mhm.
baby snakes, you have these adult snakes, and you are seeing age-related venom phenotypes. So that's something that we, certainly have to control for when we're looking at spatial patterns of variation.
And Miles asks, are there particular ecological pressures for the spitting versus biting behaviors?
Yeah, that's a great question. So the answer is definitely. I think the jury is maybe still out a little bit on some of the kind of proximate pressures that are happening in real time. There has been some really kind of outstanding work lately, looking at this on a more evolutionary time scale. And kind of the leading school of thought here is that spitting actually co-evolved with primate predators. So us, right? So spitting is a really great deterrent if something like a human or an early human can beat you with a stick or throw a rock at you. You would counter evolve, quote unquote, to propel venom at that human. So there's a lot of thinking that primates may be the main driver of the spitting behavior. So certainly that over evolutionary time, but the factors maintaining that spitting versus that biting or non-spitting maybe are a little bit more foggy.
Very cool. And let's see here. Madeline asks, is there ever sexual dimorphism in venom composition, particularly in species where males and females are very different sizes? Would they be pursuing different prey?
Yeah, that's a very interesting question. So, the bulk of the literature indicates that there is not really sexual dimorphism in venom biochemistry. I'm trying to think of like an offhand example where there might be. There are certainly examples where, male snakes and female snakes that are sexually dimorphic on the basis of size are pursuing different prey. But as far as been kind of demonstrated thus far, I don't think there's a good example of sexual dimorphism in venom composition. I guess worth noting, one of these cobras, it's the Philippine cobra, actually has a sexual dimorphism in fang morphology, which is weird and unexpected, perhaps. But certainly no, I guess, good demonstration of dimorphism in venom composition that I'm aware of.
And Phil asks, in reference to the intra-variation of venom content, do we know for sure that each individual snake produces the same venom profile with each ejection? Can it vary in the kind of prey the snake is hunting? Does it vary with the age of snake? So these are similar questions about confounding variables of lumping the whole species together, right?
Yeah, for sure. Yeah. So, yeah, great question. So, of course, we talked about age a moment ago, right? Yeah, age definitely plays a role. As far as we know, and again, kind of as far as been demonstrated, there really is no variation in the individual, kind of on a bite per bite basis. There's some really foundational work with spitting cobras, where they had the cobras spray, a pane of glass multiple times, and they ran each individual spit kind of differently. And what that at least showed is that no, there does not appear to be any difference from one spit to another or one bite to another. So we can expect individuals to be the same, provided they're kind of in adulthood. There shouldn't be much variation over like a time scale.
Yeah, great questions, everyone. These are some of these are like reviewer questions about your how you can trust the consistency of the venom. Um, yeah, this is great stuff, everybody. Thanks so much for all the questions in the chat. And thank you, Neil, for your talk. Really appreciate your participation in the seminar series. I think we all enjoyed it, seeing your time in the field. And so if there's no more questions, we will end here and we will be back next week for the Greg seminar series. Thanks so much, everyone.
Perfect. Thank you for having me. And it was great. I really enjoyed this opportunity.